Hello everybody. In this video, we will introduce about Melas syndrome. It's one of the frequent maternally inherited mitochondrial disorders. Melas is an acronym that stands for ME is mitochondrial encephalopathy, and LA is lactic acidosis, and the last is S shock-like episodes. This usually is a syndrome that arises in childhood, commonly between 2 and 15 years old, with some clinical manifestations below. The main symptoms of Melas are neurological manifestation and muscular manifestation. The first neurological manifestation include stroke-like episodes, dementia, epilepsy, recurrent headaches, hearing impairment, peripheral neuropathy. The next muscular manifestations such as exercise intolerance, muscle weakness, and motor developmental delay. And non-specific symptoms are lactic acidemia and cardiac manifestation. And another symptoms are gastrointestinal manifestation, endocrine manifestations, renal and hematological manifestations.
Hello guys, my name is Lộc. My presentation is the cause of this syndrome. Okay, let's start. One thing we all know is the mitochondria make the most of ATP, the chemical energy source that supplies most cell activity. Therefore, mitochondria are called cellular energy plants. Genetic diseases related to mitochondria are usually inherited in the motherline. Mutations in the mitochondria will affect the body energy supply and lead to many related complications. Melas syndrome is an example. Nowadays, the pathogenesis of Melas syndrome is not fully understood. The observed phenotype of Melas syndrome can be explained by several interacting mechanisms, including impaired mitochondrial energy reduction and microvascular encephalopathy, and the last is nitric oxide deficiency. Mitochondria contain their own DNA molecules called mitochondrial DNA. Gather on the chain on these mitochondrial DNA into the mitochondrial genome. The molecules of the mitochondrial genome are on circular DNA. This is the point mutation at position 3243 that is replaced by G of anti-TL1 gene. This gene encodes of tRNA leucine and before 18% of individuals with Melas syndrome. Okay, now we will talk about energy deficiency. The mutation results in the impaired mitochondrial translation that leads to impaired mitochondrial energy reduction. It makes the amount of energy reduced is not enough to meet the need of the organ, result in the multi-organ dysfunction. The next is microvascular encephalopathy. Energy deficiency can also stimulate mitochondrial proliferation. Encephalopathy due to mitochondrial proliferation in smooth muscle and endothelial cells of small blood vessels lead to the impaired blood perfusion in microvasculature, contributing significantly to the complication observed in the Melas syndrome, particularly stroke-like episodes. And the last problem we mentioned is nitric oxide deficiency. Nitric oxide deficiency occurs in Melas syndrome and can contribute significantly to its complications. This results in decreased vasomuscular perfusion, leading to limited availability of nutrients such as amino acids and thus decreased muscle protein synthesis that may contribute to the myopathy and muscle wasting. There are also some other pathogenic mechanisms that can be referenced in this field.
Regarding to treatment, there is currently no known treatment for the underlying disease, which usually progresses and fatal. Individuals are managed according to what area of the body are affected at any particular time. Enzymes, amino acids, vitamins, and antioxidants have been used, but there has been no reliable successes described. There have been no control study on long-term use of a dietary manipulation, but the below supplement has presented some promise and have offered hope to Melas individuals. Coenzyme Q10 has been helpful. Nicotinamide have been used due to complex acceptance of electrons from NADH with a ultimate transference of electrons to a coenzyme Q10. The next one is riboflavin has been described as improving the pattern functioning with complex and deficiency and 3350TC mutation. L-arginine has been used to deal with conditions. L-arginine has been administered during the series as well as the interpretal periods and might represent the probable new management for this syndrome due to diminished brain damage because of the diminished vasodilation in intercerebral artery owing to nitric oxide reduction. And one more supplement I mentioned is a short-term C2-line supplementation increase nitric oxide production to more than arginine because of the significant increase in de novo arginine synthesis associated with C2-line supplementation. Menadine-1, vitamin K3, phylloQ1, vitamin K1, and ascorbate have been used to donate electrons to a syndrome C. Independence has also been used to treat this condition. Dichloroacetate is another compound used with the agent since level of lactate are lower than plasma and cerebrospinal fluid (CSF). Reagent monohydrate has been used and increasing in muscle strength. And the last is succinate. Succinate control convulsion yet to be investigated or widely recommended. About the new treatment of biolidazones and deoxyribonucleosides combination treatment. In detail, the BioG and DNS combination treatment is read mtDNA copy number and mitochondrial mass input controls and M.3245AG mutations separate. No adverse effect on a cell proliferation. Moreover, PIOGs and GNS also insert mtDNA encoded transcript in count separate, but has the opposite effect in a mood separate, reducing the already elevated stress level.
Hello, Smith and Les. And now I will reason about method diagnosis of Melas syndrome. The first diagnosis based on clinical manifestation. The doctor will diagnose patients if they have also at least two of the following criteria is normal early psychomotor development, recurrent headache, recurrent vomiting episode. But we should note that some patients are often considerable clinical variability. Many affected patients do not fit into one particular syndrome. The last part, I will introduce the diagnostic test. The patient had to perform the following evaluation which recommend. The first is comprehensive neurological examination with cognitive assessment. The second is brain MRI. 3 is audiological and ophthalmological examination. 4 is role assessment. 5 is echocardiogram and electrocardiogram. And the last exam is screening for hypothyroidism. Thank you for listening.